So let's begin our discussion on amyloidosis. What is amyloidosis? Amyloidosis is a condition that is characterized by deposition of amyloid at extracellular sites. It is deposited outside cell, resulting in tissue damage. Now this tissue damage is not in form of some inflammatory response. It is just that this extracellular deposition, it compresses the surrounding cell and results in atrophy. So now come on to the term amyloid. What is amyloid? Let's split it. Amyloid. Amylo means starch, like. Why starch like? Because it has property which resembles starch. This amyloid term was given by Dr. Virchow. What is that property which it shares with starch? When this amyloid is exposed to iodine, it turns brown. And when it is further exposed to sulfuric acid, it gives blue purple color. This is the way starch reacts with these two components and give this color. That's why we use the term, that's why the term starch like is used for this material. But actually this is a fibrillar protein. Amyloid is a fibrillar protein which is sort of misfolded, and this protein is present along with P component and many other glycoproteins and glycosaminoglycans. So this fibrillar protein or amyloid, till date more than 40 types of fibrillar protein which form component of this amyloid have been defined. So we can say that amyloid is not a single protein, it's a group of condition characterized by different type of proteins, misfolded protein deposition, which are fibrillar. So now let's classify the amyloid protein depending on its physical property and its chemical properties. So physical properties, what does this amyloid look like? On the EM or electron microscopy, these are fibrils of indefinite length but with a diameter of 7 to 10 nanometer or 70 to 100 angstrom. On X-ray spectroscopy, it has a beta pleated sheet structure. It has a beta pleated sheet configuration. And chemically, this amyloid is composed of two components, 95% of which is a fibrillar protein along with 5% of another component which we call as P component. So now let's classify this amyloid depending on clinical presentation. So this amyloid may be either locally deposited, so we'll call it localized amyloidosis, or this could be systemic amyloidosis, and the systemic amyloidosis is further divided into primary amyloidosis, which may be idiopathic or could be due to plasma cell disorders. Plasma cell disorders, so with this we can say that this amyloid is of AL type. They secrete, these plasma cells, they secrete light chain immunoglobulin, light chain like kappa and lambda. So AL type of amyloid, AL stands for amyloid light chain, is seen in primary amyloidosis. The second type is secondary amyloidosis. The secondary amyloidosis is usually secondary to some chronic inflammatory disorder. Some chronic inflammation, some chronic systemic inflammation. So after chronic inflammation, what happens is the liver gets stimulated and the hepatocytes, they secrete serum amyloid associated protein, which is a large molecular protein, and in the plasma it is broken down and it forms amyloid associated protein which gets deposited. So in the primary amyloidosis it is AL type of amyloid, while in the secondary amyloidosis it is AA type of amyloid protein. Other amyloidosis, which one more category I'll add in this one, is familial amyloidosis. In the familial amyloidosis comes example of senile systemic amyloidosis, which is characterized by deposition of transporter of thyroxine and retinol. Transporter of thyroxine and retinol. It's a normal protein. When this normal protein gets deposited, we call it senile systemic amyloidosis. But when this protein is mutated, we call it as amyloid TTR, amyloid transporter of thyroxine and retinol, and it gets deposited, then we call it as familial neuropathic amyloidosis. And this is the normal protein and this is a mutant protein. Other examples include amyloid beta type, which is found in Alzheimer's disease. One more type, we have beta 2 microglobulin type of amyloid, and it is found in patients who are frequently which underwent frequent hemodialysis. So for diagnosis for the diagnosis of amyloidosis, the definitive diagnosis is attained only on biopsy, only on demonstration of amyloid on histologic section. Histologic section of the biopsy tissue. So from which sites we can do the biopsy? First is abdominal fat pad biopsy, and we can do from rectum as well and gingiva also. So on the biopsy, what do we see? What all special stains we'll be using? So on HE stain or hematoxylin and eosin stain, what this amyloid will look like? It's an amorphous hyaline deposition like material deposition in extracellular sites. With Congo red stain, it will give pinkish red color on light microscopy, but on polarizing microscope it will give apple green birefringence. It looks beautiful on the polarizing microscope. And with the methyl violet, it will be metachromatic. And plus you can always put an immunohistochemistry for SAA in cases of secondary amyloidosis. One more thing I would like to discuss, this protein, where does this amyloid protein comes from? What are the pathogenetic mechanism behind this protein component which is misfolded? Protein component, this fibrillar protein component which is misfolded, from where does this component comes from? This could come from either there is a secretion of a normal protein in excess amount. So since it was being secreted in excess amount and it was more prone to get misfolded, so it misfolds. Another mechanism is that the protein is secreted after mutation. So the mutant protein, which is more prone to misfold, forms misfolded protein. One more important thing behind the accumulation of this misfolded protein is that there is decreased degradation as well. There is decreased degradation of this misfolded protein. Obviously then only this is going to accumulate in the tissue. Thank you.